So thank you for joining, all of you, and welcome to our October monthly seminar for the Superfund Research Program at Columbia University. And this seminar, it's a really wonderful interdisciplinary event where biomedical scientists and earth scientists come together and discuss scientific questions and issues of great importance, not only for science, but for also for action and policy and communities related in general to metal exposures, and arsenic, with a very strong focus on arsenic. And today we have two wonderful speakers. Both of them are, launching their careers. They are really doing amazing work. I think it's exciting for us, for our program, to see both of you, Arte and Craig, who are our speakers today, doing really amazing, fascinating work. I think it gives us a tremendous hope that we are in the right direction, that this is really, really exciting. And so I am going to start introducing Arte Domingo. So Arce is a biostatistician working with us at the Columbia University Superfund Research Program in the Department of Environmental Health Sciences. She got a BA in mathematics. And after that, she specialized in biostatistics with a Master of Science in Biostatistics from the University of Valencia, and her degree in mathematics was from the Autonomous University of Madrid. Following her master's, she came to Columbia University and she started working with us. And then she went back to Spain and she continues still working with us, but she started her PhD in biostatistics, both at the National Center for Epidemiology in Spain, which has been very, very busy, as you can imagine, lately with COVID activities, and also continuing her training also with the University of Valencia. And she's going to be sharing with you a lot of the work that she has been really moving forward related to metal epigenetics and chronic disease outcomes in the Stronghold study. So Arce, thank you so much for joining us today. I know that it's quite late for you in Spain, so we are really privileged to be able to hear directly from you. And thank you to all of you for joining. If you have questions, I was thinking you can put them in the chat, and then at the end, Arte can answer them and go over them. And then we'll open the floor also for open questions if people want. Okay, thank you.
Thank you, Anna, so much for the introduction, and thank you all for joining the seminar. It's a great pleasure to be a speaker in this seminar, in which I have learned so much in the past from experts of different fields and from very rich interdisciplinary discussions. First of all, I have to say I can see many familiar faces, and I know many of you have heard about my work and about this project. So I apologize if you have to hear many times about the same projects. And let me share my screen. Okay. Great. So I'm going to talk about metal-induced epigenetic changes in chronic disease. Now that omics data have become such an important topic on the attempt to disentangle how exposures cause disease, it is our challenge to figure out how to best analyze, interpret, and take advantage of this data. And to do so, we need powerful data from long and well-designed studies which enable the growth and richness of epidemiology. So I'm sure many of you know the Stronghold study, but for those of you who don't, I'd like to introduce you to this population-based prospective cohort, which is the largest study of cardiovascular disease in American Indians ever undertaken. This study is supported by the National Heart, Lung and Blood Institute and the National Institute of Environmental Health Sciences. It includes 12 American Indian tribes from Arizona, Oklahoma, North Dakota, and South Dakota, with more than 4,500 participants. And all the investigators involved in this study do a community-engaged work, which means that every research item needs to be proposed and evaluated by the tribes so that we can get feedback and suggestions from them, and that way achieve better science. Participants were followed up for almost 30 years for several outcomes, including cancer and cardiovascular disease. The reason why cardiovascular disease was the main aim of this study is that American Indians suffer a disproportionate burden of cardiovascular disease as compared to other US populations. For example, we can see in this chart the rate of coronary heart disease of the Stronghold study as compared to that of the ERIC cohort, also from the US. So we can see that it's much higher, and unfortunately, this is very sad for the communities, but at the same time, it provides a unique opportunity to study cardiovascular disease from the epidemiologic point of view. Looking at environmental exposures that might explain part of the risk of cardiovascular disease and other chronic diseases, metal exposure is a global public health concern, which have been related to adverse health effects. Metal exposure in the Stronghold study is low to moderate. However, it's still higher than some other US populations, such as the multi-ethnic study of atherosclerosis, as we can see in this chart. Arsenic and cadmium are some of the most well-studied metals in terms of adverse health effects. Cadmium has been associated to cardiovascular disease, increased blood pressure, and cancer mortality in the Stronghold study. And also arsenic has been associated to cardiovascular disease, cancer mortality, restrictive lung disease, diabetes, and chronic kidney disease in the Stronghold study. However, the biological mechanism by which these metals might contribute to disease is still unclear. One of these biological mechanisms might be epigenetic marks. Humans are made of more than 200 different cell types, and each cell has the same DNA. Epigenetic marks are responsible for turning genes on and off so that cells can have different functions, so they induce phenotype changes that do not alter the DNA sequence. A clear example are twins. They have exactly the same genetic code, but they sometimes have different physical characteristics, as can be seen in the photograph. So epigenetic marks are responsible for these changes. Now, the interest in epigenetics has grown a lot in the past years, mainly due to three different reasons. The first one is that they have been proposed as a causal mechanism for disease. For example, epigenetic dysregulations in the AHRR gene induced by smoking have been proposed to have a causal role on the development of lung cancer. They have also been proposed as a biomarker for disease prediction. For example, epigenetic dysregulations sometimes happen long before oncogenic transformations start in cancer, so they might be helpful for early detection of disease as well. And last, pharmaceutical industries have been investigating epigenetic marks as therapeutic targets, and some of them have already been approved, for example, for leukemia. But for us as epidemiologists, the first two bullets are definitely the most relevant ones. The most broadly studied epigenetic mark is DNA methylation, which happens when methyl groups are added to the DNA molecule. This mechanism is essential for normal development and associated with key cellular processes. However, given that it affects gene expression, these regulations might lead to overexpression or underexpression of certain genes. Environmental exposures are known to highly influence DNA methylation. In fact, most of environmental exposures cannot alter DNA sequence, but they can alter the epigenome. The impact in global methylation is thought to happen by direct impact on the function of DNA methyltransferases and enzymes, which are responsible for methylation and demethylation. But the impact in gene-specific alterations is less clear. One of the hypotheses is the transcription factor occupancy theory, by which environmental contaminants would influence transcription factors and that way induce dysregulations in methylation. So given all this evidence, we are interested in studying the role of DNA methylation changes on the association between environmental exposures and chronic diseases. Regarding environmental exposures, we have focused on cadmium and arsenic, which, as I said, have plenty of evidence of being related to adverse health effects, and smoking, which, as you all know, is the leading cause of preventable death in the world. And regarding chronic diseases, for the moment, we have focused on cancer, cardiovascular disease, and diabetes. So in this slide, we have 4 different ongoing projects, and I'm going to give you a little overview of each of them during this talk. Since all the projects involve DNA methylation, I'm going to briefly explain how these data were preprocessed in the Stronghold study. DNA methylation was measured in 2,351 participants at the baseline visit using Illumina's methylation EPIC bed chip, or 850K. Samples with low intensities, non-bimodal distributions, CpGs that were technical failures, sex chromosomes, cross-hybridizing probes, and SNPs with minor allele frequency greater than 0.05 were excluded. And we applied 2 different normalizations, single sample normalization for background correction and regression in correlated probes normalization for probe type bias. Finally, we also did a batch effect correction. And after all these preprocessing and exclusions, we had 2,325 participants, as well as almost 800,000 CpGs for the analysis. I'm going to start with project 1, which involves cadmium, smoking, and DNA methylation. Now, cadmium is one of the main toxicants in tobacco, and as I said before, it's associated with smoking-related health outcomes, such as cardiovascular disease and cancer. It has also been associated with changes in global DNA methylation. However, to our knowledge, no epigenome-wide association study in blood was published prior to this work. So the objective of this study was to investigate the potential mediating role of cadmium on the association between smoking and DNA methylation. To do so, we first did an epigenome-wide association study to look for differentially methylated genomic positions, or DMPs, for both cadmium and smoking. And then we did a mediation analysis using the beta regression for proportion data, as well as the product of coefficients method. To summarize the results, here I'm showing the number of differentially methylated positions for cadmium, current smoking, former smoking, and cigarette pack years or smoking intensity. We can see that current smoking has much more DMPs than the others. For cadmium, we found 6 differentially methylated positions, and 4 of them were still significant when restricting the analysis to never smokers. And for current smoking, we replicated the association of well-known genes such as AHRR and F2RL3, but we also found some new genes that were not reported before, such as this last one, which is bolded. Regarding the mediation analysis, here I'm showing the total effect of smoking on DNA methylation, the mediated effect through cadmium, and the relative mediated effect, which is the percentage of the total effect attributable to the mediator, in this case, to cadmium. So here we can see we have up to 13% of relative mediated effect. As conclusions, we replicated the association of smoking-related genes and found new ones that were not reported before. Cadmium was associated with changes in DNA methylation, even among never smokers. And the effect of smoking on DNA methylation might be partially mediated by cadmium. However, most of the effect is still unexplained. So further research is needed regarding other toxicants of tobacco. And here's the paper, which was published in Environmental Health Perspectives, in case someone wants to read it. Moving forward to project 2 about smoking, DNA methylation, and cancer. Smoking is associated to 14 different types of cancer, and 36% of smoking-related deaths are for cancer. Also, 80 to 90% of lung cancer deaths are attributable to smoking. And one of the possible biological mechanisms by which smoking might induce lung cancer is DNA methylation, as we know that it can change healthy regulation of gene expression to a disease pattern, and DNA methylation changes occur in early stages of tumorigenesis. So the objective of this study was to investigate the potential mediating role of DNA methylation on the association between smoking and lung cancer. And to do so, we first did an epigenome-wide screening to select the CpG sites that were related to lung cancer. In the previous project, we considered CpG sites one by one. However, due to the high correlations and huge confounding between CpGs, studying them all together might be more appropriate. So that's why we used a modification of the popular ElasticNet tool, Adaptive ElasticNet, which incorporates data-driven weights in order to achieve better consistency in estimation. Then we applied mediation analysis only to the selected CpGs using Allen additive hazard models, which are a quite standard tool for mediation analysis in a survival setting. For current smoking, we found significant mediated effects for 9 CpGs, and here I'm showing the top 6. So here the total effect is quantified as number of cases of lung cancer attributable to smoking per 100,000 person years. The mediated effect is quantified as the number of cancer cases attributable to smoking through DNA methylation. And again, the relative mediated effect is the percentage of the total effect attributable to the mediator, in this case to DNA methylation. So here we can see that we have quite high relative mediated effects. This would mean that 72% of the effect of smoking in lung cancer could be explained by changes in DNA methylation in this gene, which, as I said before, is a very well-known smoking-related gene, AHRR. As conclusions, a large extent of the effect of smoking in lung cancer might be mediated by DNA methylation changes in well-known smoking-related genes. However, these results need to be interpreted with caution, as we are considering cancer incidence as the cancer diagnosis date, but we know that these oncogenic transformations might have been ongoing long before this diagnosis date. So the biological interpretation here is quite complicated. Also, since we have self-reported smoking data, we might be subject to exposure misclassification. However, we did a sensitivity analysis considering self-reported smoking exposure in 2 different phases of the study, and we excluded participants with discordant smoking status. We obtained very similar results after that. Moving forward to project 3, arsenic, DNA methylation, and cardiovascular disease. Arsenic is a toxic metalloid that mainly comes from water, food, soil, and air. And in 2019, it was the #1 toxicant in the Agency for Toxic Substances and Disease Registry Substance Priority List. High chronic arsenic exposure has been linked to cardiovascular disease in populations from Taiwan, Chile, and Bangladesh. And also low to moderate exposure has been linked to cardiovascular disease in several populations, including American Indians from the Stronghold study. This evidence is also supported by experimental studies in mice. Inflammation, oxidative stress, and cardiovascular disease risk factors are some of the proposed mechanisms for this association. However, emerging evidence also suggests that epigenetic mechanisms might also be part of this biological link. Arsenic exposure has been associated with changes in global and site-specific DNA methylation, including the Stronghold study, and also changes in DNA methylation have been associated with cardiovascular disease and its risk factors. So given all this evidence, we were interested in looking at the potential mediating role of DNA methylation on the association between arsenic and cardiovascular disease. And to do so, we used the same methods as in the previous project. So we did first an epigenome-wide screening to select CpG sites that were associated to cardiovascular disease. And then we applied mediation analysis to those selected CpGs using Allen additive hazard models. Here I'm showing the results for the mediation analysis. We found 9 CpGs with significant mediated effects. And here I'm showing the top 6. The total effect, mediated effect, and relative mediated effect are quantified exactly the same way as in the previous project. And here we can see we have up to 18 relative mediated effect. These genes have very interesting biological functions. In fact, 2 of them have been proposed as therapeutic targets for cardiovascular disease. 3 of them have previously been associated to cardiovascular disease and its risk factors, including one of them having also evidence from experimental studies. And very importantly, 3 of them have been associated to diabetes, including this last one, which is an autophagy-related gene. Autophagy upregulation is a possible mechanism for arsenic-induced diabetes, as published in this study. So this tells us that diabetes might be an important mechanism in arsenic-induced cardiovascular disease. As conclusions, DNA methylation might be a partial mediator on the association between arsenic and cardiovascular disease. Since 3 out of the 9 significant genes were diabetes-related, diabetes might be an important mechanism for arsenic-induced cardiovascular disease. And also experimental studies are needed to assess whether changes in these epigenetic signatures in arsenic-exposed animals influence cardiovascular disease development. So moving forward to the last project, which is DNA methylation and incident diabetes. American Indians and Alaska Natives are more likely to develop diabetes than any other US racial group, according to the Center for Disease Control and Prevention. And studying cardiovascular disease risk factors in these populations is particularly important given the high burden of cardiovascular disease that they suffer. DNA methylation has been reported to play a role in the pathogenesis of diabetes. So in this study, we were interested in looking at the association between DNA methylation and diabetes in this population. To do so, we developed a 2-stages method. In the first stage, we did a cross-sectional analysis looking at the association of both fasting glucose and insulin resistance, or HOMA-IR, with DNA methylation. And then in stage 2, we did a survival analysis for incident diabetes only in the selected CpGs, using participants that were followed up between 1989 and 1999, so in visits 1 to 3 of the Stronghold study. And participants with prevalent diabetes were excluded for this analysis. So this work is still at a very preliminary stage, but as preliminary results, we can say we found 22 CpGs related to fasting glucose and 78 related to HOMA-IR. However, when adjusting the model for BMI, no CpGs remain significant. When doing the incident diabetes analysis on the selected CpGs, 20 were associated with incident diabetes, going down to 10 when adjusting the models for BMI. Importantly, the 2 top genes have been proposed as biomarkers for type 2 diabetes risk prediction. Also, in this study, we want to look at the potential mediating role of DNA methylation on the association between arsenic and diabetes, as we previously did for cardiovascular disease. So I wanted to end with some general conclusions and potential future work. DNA methylation might be part of the biological link between environmental exposures and chronic diseases, and might also be a good and non-invasive biomarker for early detection of disease. However, one of the main challenges is to disentangle whether these differentially methylated positions and regions are actually part of a causal mechanism or just exposure biomarkers. As I said, both hypotheses have been proposed. So some of the mediation signals might just be very good exposure biomarkers rather than being on the causal pathway. So experimental studies in animals are needed to assess this question, and this will be one of our next steps in the Stronghold study. Another next step would be to incorporate multi-omics data, since at the moment we only have DNA methylation data, but we are planning to incorporate other omics data so that we can look at this association from a more broad perspective. Another potential next step is to try to estimate the heritability of these environment-induced epigenetic dysregulations. And in this line, an extension of the Stronghold study called the Stronghold family study was created, first as a pilot in phase 3, and then fully incorporated in phase 4. And this was a family study, as I said, which included SIP-ships with at least 3 Stronghold study participants, which are the black shadings in this figure, 5 or more siblings, black and diagonal shadings in the picture, and at least 12 offsprings, which are the dotted shading parts of the Stronghold study participants. So as you can see, this study includes a lot of families with many generations. So this is a very rich study and the ideal setting to do these heritability estimation studies. So I wanted to acknowledge all the people that have been part of this study. Of course, the Stronghold study participants and investigators and all of the collaborators from different universities and research centers. And of course, also our funding and Columbia Superfund research program. So I'll be happy to take any questions now, but please also feel free to email me in case you have questions or suggestions or potential collaboration proposals, and I'll be very happy to discuss. And thank you so much for your attention.
Okay, thank you. Thank you, Arce. And maybe you can stop sharing so that we can see each other's faces. And you know, you went quite fast, so we have like plenty of time for questions and discussion. So I think this is great, because I think there is what you've presented. It's actually quite complex, and there is a lot of complexities underneath. So it's great that we have time for discussion. And there are already several questions out there. Can you see them?
I can see them. So Sadija Tashnia is asking, apart from the smokers and non-smokers, is there any impact on the person who is exposed to the smoke inhalation but not directly involved in smoking? So this should be addressed using secondhand smoke data. We do have some data of secondhand smoking exposure in the Stronghold study, but it's not, I think, well, Anna, you might explain better, but it's not as well quantified as we would like it to be. And we actually did an analysis with these secondhand smoking data, and we did not find any clear associations with the methylation data. But definitely, I mean, for sure, this has an impact in DNA methylation, but we have not been able for the moment to quantify it in any way. And then Andres Cardenas is asking, I wonder if Linda Valeri has some feedback on using DNA methylation as statistical mediators, as she has shown that DNA methylation might be a better biomarker of exposure than self-reported smoking, as well as suggestions to overcome this issue. Linda, do you?
Hi, I mean, I think, thank you, Andres, for the question, but I think, Arce, you actually mentioned this in your presentation towards the end as one of the challenges. I think, Andres, you're mentioning that paper in Epigenomics. So we explored in the MOBA cohort the mechanism of smoking effects on birth weight, and DNA methylation was again a mediator there. And I think that in our study, we understood that it's much better than using continuous measures of the exposure than categorical, because if you have a continuous mediator very precisely measured, that you know can be used as a proxy actually for the exposure, whereas the exposure is very crudely measured, only categorical, then just because of this imbalance of an imprecise categorical exposure and the continuous mediator, you might find correlations that are not, basically the big issue is the causal effect of DNA methylation on the outcome. And if you adjust for smoking, if smoking is imprecisely measured, DNA methylation captures some of that effects. So it's basically might steal some of the causal effect of smoking just because it's a good proxy for it. So the answer to this problem is on exposure science. That is, how best can you measure this exposure? Can you capture it at the time where the development of the disease, is happening, and it's very difficult. So the answer here is better the study designs, intense longitudinal data on the exposure whenever possible, and in addition, sensitivity analysis for measurement error, for which there are several options available. But it's a difficult problem. Albeit, I actually think that these results are really causal. It's just that is, in our sensitivity analysis, we could not explain away the mediating effect due to measurement error, but in fact, if you see that sometimes if you look at each CPG sites together, you might find proportion mediated, that they sum up to more than 100%. That's already a red flag. That is either due to measurement error bias or inappropriately accounting for the correlation among the CPG sites. So there are multiple aspects, but it's great work, Arce. I think that you're doing a top-notch analysis for the data and the design that you have available to conduct this study.
Thank you. And Linda, do you have any recommendation regarding, since, as you said, they are not independent, the CPG sites, in fact, they are correlated and they are adding up to more than 100%. Do you think a multimediator model would be feasible here in terms of, obviously they are not independent, so it's a complicated issue. As I have read in the literature, being intertwined like these mediated pathways is an issue to do this multimediator analysis, but do you think this is feasible in this context, or?
I think it depends on your causal question. That is, if your goal is to identify the drivers of you can always do a joint mediated analysis through all the mediators together, but then it wouldn't address the question which of these CPG sites are more important in this mechanism. So I think that it's a mix between causal discovery and integrating in your model what is known about the biology. So, for example, we are starting to see how network structures of the gene could be incorporated as mediators. So, for trying as much as possible to maybe group genes that belong to the same pathway, so that maybe there is the inflammation pathway, there is the, I'm not you are the expert here, but maybe you can identify how many the genes that work together in explaining the underlying pathway. Maybe is either inflammatory or is the cell production, or again, maybe you can use some of the biology to group them. And more automated way that would be more statistical and less causal could also be used as well as exploratory tools. But
there's
a lot of research going on.
It's definitely
and
challenging.
And you can even contribute too with some new methods for your PhD.
And Maria Telles has a comment that is kind of related. that
Yes,
does
Maria.
it make sense? Yeah, you can read it, Arce.
Yes, Maria Tellez is saying, does it make sense to talk about statistical mediation? Mediation relies upon the hypothesis that the mediator outcome relation is causal. So if you do not believe it is causal, you do not conduct mediation analysis to begin with. Yes, it might be like a kind of abuse of statistical notation, I guess, since we don't know if this relationship is causal or not. Yeah, I mean, I don't know.
And Andres, maybe do you have something else that you would like to add to this discussion related to what Linda said?
Well, I wanted to thank Arce. That was a really nice presentation. And I actually, as I finished typing that, I think the next slide, that's when you talked about the exposure misclassification issue. So I apologize, maybe I should have waited. And Linda, I appreciate your feedback too. I was wondering about testing for exposure mediator interaction, because maybe here we're making the assumption that smoking doesn't really interact with methylation, but maybe the methylation levels might be, potentially interacting with exposure, and whether you tested those. And also maybe, you can speak or Linda about the potential for exposure mediator interaction here with methylation and smoking at least.
Yeah, I mean, I haven't tested any interactions between smoking and DNA methylations. What we did is, actually I was asked from a reviewer in EHP. There is a tool called Epismoker, which quantifies, is able apparently to determine smoking status from DNA methylation data. So we run that to see if actually smoking status could be predicted from methylation data in our study. And actually, for former smoking, it was not a good prediction in our data. So this might be related to the fact that methylation marks might, these methylation changes might disappear with time upon smoking cessation. So I'm not sure about interactions, but this might be a relevant issue to take into account, especially when conducting mediation analysis, since there are these, statistical assumptions that need to be fulfilled about this exposure mediator relationship and exposure mediator outcome relationship and so on. So this might be relevant to test, but I haven't done any work on that, on interactions between exposure and mediator in this setting. But I think it's important to quantify, as Maria said, that this mediation analysis has a lot of assumptions that are not easily fulfilled. It's very important to do, as you said, interactions or sensitivity analysis of the exposure mediator relationships to be sure that we are not, that we are kind of fulfilling the statistical assumptions required before taking any conclusions from this study. That's true.
There is a question, from Steve Chilrod.
Yes. So great overview of 4 separate studies, but it went very fast for a geochemist. I'm so sorry for that. And studies that have focused on the genetics of lung cancer tumors used for determining targeted therapeutics has suggested a larger fraction of lung cancer not to be associated with smoking, even in smokers. Can your approach help find other mechanisms for lung cancer for non-smokers or smokers that don't get the smoking type of lung cancer tumors? So do you mean DNA methylation related lung cancers that are not due to smoking?
So, this is a very personal question. My wife had 30 pack years, but the genetics of her cancer was not consistent with smoking. So I'm asking whether your approach can not only be looking into smoking, but can it look into non-smoking cancer types and see what might be the mechanism for those cancers?
Yes, for that, we should do an association analysis between just DNA methylation and lung cancer, and maybe look at the biological pathways of the differentially methylated positions to see, which pathways are related to this lung cancer. But this, for that, if it's a genetic thing, we would need genetic data also, another omics data. So I have heard that, however, for lung cancer, I'm not sure if this is true. I know nothing about the real biology, of lung cancer, but I had heard before that only 10% of lung cancers are explained by genetics. So if this is true, most of the lung cancer might be explained by, epigenetic changes or other biological mechanisms that are not genetic. So, but yeah, this should be tested looking at biological pathways related to this differential methylation. But in this study, since I have only focused on, the smoking, DNA methylation and lung cancer relationship, I haven't done any work on, other potential sources or exposures or mechanisms that might induce lung cancer.
Right. I guess part of it was also in your analysis, you assume if someone identified as a smoker, mm-hmm. that fell into your smoker bin, right? And what I'm saying is, at least in my experience, the doctors told us, oh yeah, you were a smoker, but that had nothing to do with your lung cancer. Mm-hmm. So that bin of calling people smokers has some variability in the types of lung cancer that you get. Mm-hmm. Some are apparently associated with or caused by the smoking, and some are not, which I was shocked at, but so be it. You learn something. And so your analysis is assuming that everyone who self-identifies as a smoker, you put them in your smoking bin. Some of the variability you saw might be due to the type of lung cancer that they had. Because what was also very evident is that there's many types of lung cancer. There's not just one type of lung cancer. Otherwise, these targeted therapeutics would work on everyone, and they don't. They work on all these different subsets of people.
Yeah, totally. Yeah, that's a great suggestion.
I think the limitations we have here is that we don't have enough power for these different subtypes because the number of events is limited. And so to run those analyses would be, I would say, very complicated with the data that we have. But trying to see signals, trying to see differential signals, I think in Arce's work, clearly there is very strong DNA methylation signatures that are related to smoking. Either they are causal or they capture very well the smoking's effect, one or the other, through direct mechanisms, causal mechanisms, or just indirectly by capturing that relationship. That's very clear. But there are others. There are other mechanisms that could be captured through DNA methylation independent of smoking. Yes. And actually, in another work that Arte has done, she has looked at the effect of DNA methylation per se independent of smoking after you adjust for smoking. And yes, you can see DNA methylation signatures that are independent of smoking status. So that answers your question in a way that yes, there are other DNA methylation signatures that are not related to smoking and that are still related to disease in this case.
Okay, that's what I was looking for. Okay.
Very good. So Ben has another question, and I think we can go over for 5-10 more minutes, and then we'll get a break before we go to the next talk. But Ben has a question. Ben,
yes.
Bostic, yeah.
Maybe a bit of a random question, but are there any environmental factors that affect DNA methylation more broadly? And if there are some, would that relatively uniform suppression be distinguished from effect on a specific gene? Yes, definitely. In fact, what is clear is that environmental exposures affect global DNA methylation, and actually the biological mechanism is, more clear, as I said before, that they have a direct effect on DNA methyltransferases and these enzymes that do methylation and demethylation. So it's quite understood the role of environmental exposures on global DNA methylation. So what is more challenging is the gene-specific type So that's where there are several hypotheses on how that could happen. And yeah, definitely it can be a different biological mechanism than that for global DNA methylation, that for site-specific DNA methylation in terms of environmental exposures. And then Maria has another question. No, the mediation package does exposure mediation interaction testing, I recall, only for a limited amount of outcome types, definitely not allen additive hazards models in R.
I think, yeah, this is a conversation between with Andres, and Andres says that it can be done. Yeah.
Okay. Mm-hmm. And then
Brandon is actually responding to Steve. So thank you, Brandon, for responding to Steve. And I think that's a very important point, that at the individual level, these signatures are very complicated. What we are capturing here are signatures at the population level. So moving from the population level to the individual level is always very, very difficult. So Karen is asking, is there a way to look at whether diet modulates the DNA methylation?
Yes, there are some studies looking at diet, but well, since diet influences so much BMI, for example, BMI, the relationship with BMI is complicated. It's unclear whether it's BMI what has an effect on DNA methylation or the opposite. DNA methylation has an effect on BMI. So since diet influences BMI, in case BMI has an effect on DNA methylation, this would definitely be related. But the way in which you would look at that, I mean, it's definitely self-reported diet in people. And I haven't looked at any particular studies with diet in DNA methylation, but I know there are people that have done some work, and there are some evidences that, of course, diet, as any other, external exposure, influences DNA methylation. But again,
yeah. I was thinking about it, like just looking at just the top hits correlating arsenic and CBD, just the next to last vignette. If you look at the genes that you've picked up, going back to sort of what Linda was going for as far as pathways, there's not really enough genes to think about pathways at this point, at least from my, from the experimental animal perspective side of this. I wouldn't even attempt to try and understand the modeling that you've done. That is beyond what I'm familiar with. But if you just look at the genes that have come up, they're not super similar to each other. You have TGF-beta receptor that's involved in a lot of important transitionings and developments. You have collagen, which I think that may be, if I were to pick something of the list, that might be the one I would go first in an animal model, because I think that one's the most intriguing as far as the fibrotic phenotypes that it could lead to. But the other ones, you're dealing with genes that are involved in thymidine phosphorylation or an E3 ubiquitin ligase. These seem like so variety, like it's kind of a variety. I'm not sure what pathway I would put that all into. So I guess I was wondering if you could take any other factor and narrow it down to what you might think is causal that way. And diet just came up.
First of all, these genes, in this kind of analysis, you wouldn't get like correlated CPGs. I mean, they still might have, similar biological functions, but correlated genes are not present since we have done this screening to select CPG sites, and always these shrinkage tools tend to not select very correlated or CPG sites. So that for pathway analysis is not ideal. I mean, for pathway analysis, it might be better an individual-based analysis in terms of you're going to consider a lot of CPGs and look at which pathways are enriched or whatever. And also regarding diet, since you are relating it to arsenic, to the arsenic study, in this population, the main source of arsenic is water. However, there are some tribes in Oklahoma that might have diet as the main source of arsenic since they have a low exposure from water. So diet might also be relevant, but I see your point, like looking at other like sources or like more specific things to narrow it and have a more like clear perspective of where to go with the
example,
study?
One thing you might do, just sort of, I'm sort of making this up, but if you looked at whether selenium changes any of those specific ones, could you narrow down if you think that the selenium levels, if they're reasonably high, could combat a high arsenic, does that modulate something? Would you lose an effect somewhere? So sort of looking at an intervention without actually doing an intervention. You know what? Maybe I'm not making myself clear. Anna can translate my animal-ish.
Yeah, I think it's a very interesting thought. It's kind of similar, Arce, to what happens in the diabetes model when you adjust for BMI. The association goes away. And so here you could see if the association between arsenic and DNA methylation in the causal pathway to cardiovascular disease, if it's causal, could it be modulated by some other nutritional or environmental factors, for instance, selenium? And if selenium has any potential beneficial effect, or maybe not beneficial, but maybe we could see it that these relationships go away and disappear. And so that could help us to disentangle, I don't know, potentially, whether we are talking about some biomarker signatures versus some real mediation effects on the outcome. Something I was going to say also related to that is that for the smoking findings, I see that a lot of the potentially mediating CPGs are really biomarker effect exposure assessment related CPGs. That's what I see from what you've shown, because these CPGs have really primarily been related to the exposure. That's what we know about them. However, for the arsenic cardiovascular disease mediation analysis, the types of CPGs that were selected seem different. They seem to be more related to diabetes, to outcomes, to different pathways. They don't seem to be the ones that we found with Anne Bozak's analysis that were related to arsenic exposure per se. So it's a very, very different picture. So I think those types of differential findings might help us to kind of, at the end, conclude if our hypothesis is that primarily these CPGs are primarily biomarker related, exposure assessment related. I think that's the case for the smoking findings, I think. While for the arsenic outcome findings, they seem to be something else. And so that, of course, we are limited in our epidemiological research. This is observational, this is hypothesis generating. I think it's very complicated to look at mechanistic pathways in epidemiological studies. And I think you need to pair this with experimental models. Yeah, sure. Otherwise, we are not going to be able. But if these pathways, some of these can be confirmed, that could be quite potentially interesting. And going back to Corin's question about that the pathways are so different, that is related in part to what Arce mentioned, that the statistical methods that we are using are prioritizing the best CPG for each potential relevant pathway, because it's looking for the very complex interrelationships. And from the most related pathways, it's selecting the top CPG. And what's interesting is that these seem to be biologically relevant, while with other methods, we would select many, many, many different CPGs, but they were more difficult to interpret too. But I think we are still far away from understanding. I feel we've made a lot of progress in this DNA methylation analysis, but I think in 5 to 10 years, we'll go back to this data and we'll say, oh, now we understand what we were seeing. But we are not there yet.
Well, hopefully we'll have the animal data to help you reasonably soon. We have the initial male mice the data's back. We're just crunching the numbers. So maybe that will help.
I am sure. And thank you. Yes. Thank you, Karen. And thank you, Arce, so much for your presentation and your talk. I think we have 5 minutes. If anybody has any other comment or question, please feel free to do so. We'll just take a couple of minutes break if people need to relax a little bit or step up for a second, for a few minutes, and we'll move to our next talk starting at 4 p.m. So thank you, everybody. Thank you, Arce, especially. Thank you for the
Thank you all for your questions and suggestions. It's been a great discussion. Thank you.